In this tutorial, we are going to learn about the enzyme action. Enzyme action, the breaking of substrate into end product by enzyme, is called enzyme action. Here, the substrate breaks by the action of the enzyme and forms the end product. This theory was proposed by Michaelis and Mentine. Enzyme action involved in 4 steps. Step 1. The enzyme molecule combines with substrate molecules, forms enzyme substrate complex. E plus S forms ES. E is the enzyme molecule, S is the substrate molecule, and ES is the enzyme substrate complex. It is also known as Michaelis complex. Step 2. Enzyme contains specific site for the attachment of substrate molecule. Active sites contains the amino acid residues. On the screen you can see the image of the enzyme molecule and the substrate molecule, and the enzyme molecule has the specific active site where the substrate is going to attach. Step 3. The active sites loosen the chemical bonds into substrate, and this leads to the breaking of substrate into end product. E and S is the enzyme substrate complex, E is the enzyme molecule, and P is the end product. Step 4. The enzyme is now free to combine with another molecule of substrate. On the screen you can see the substrate molecule and the enzyme molecule gets combined and forms enzyme substrate complex, and the active sites loosen the chemical bonds into substrate, and this leads to the breaking of substrate into the end product. Here, 2 hypotheses was proposed to explain the mechanism of formation of enzyme. And it is lock and key hypothesis, and another is induced fit hypothesis. We will learn that in another tutorial. Click the I button for the lock and key hypothesis and induced fit hypothesis. If you found this tutorial helpful, then do like, share and subscribe to the channel for further tutorials.